Good morning from New York. I'm Dave Miklas, the Executive Director and founder of the DNA Learning Center at Cold Spring Harbor Laboratory. It's 7 a.m. here, and I'm happy to introduce the second in our series of great experiments in molecular biology and biotechnology. Today's experiment is restriction analysis, and we're presenting it today to show solidarity with our friends and colleagues in China as you face the threat of the coronavirus. We hope that this will give you one more great thing and happy thing to do while you're at home. Restriction analysis is one of the key techniques in molecular biology. It's really the key to manipulating DNA, and it shows that DNA behaves in a way that Watson and Crick predicted when they discovered its structure in 1953. I became interested in this experiment about 40 years ago when I was working at Cold Spring Harbor Laboratory, and I met this fella on the left who is a postdoctoral fellow named Greg Fryer. And he showed me the experiment that you'll do today, and I was astounded when I had the opportunity for the first time to see DNA and to understand how it behaves. You're going to do that same experiment today. But it's important also to know that this experiment came from the home of molecular biology, and in particular of the restriction enzymes that you'll be using today. At the time I developed this experiment, I was working in the lab of Rich Roberts. Rich Roberts, here he is at that time, he went on to win the Nobel Prize in physiology or medicine for techniques that he later did at Cold Spring Harbor. But at the time, he was the leader in discovering restriction enzymes, the very tool that you'll learn how to use today. So I want you to know that this experiment is not only being broadcast to you from the DNA Learning Center of Cold Spring Harbor Laboratory, but it also virtually came from this place as a molecular technique. I'm also really happy because today you will be taught this technique by my friend Michael Paul. And I say friend because I have known Michael since he was 2 years old, and I was very happy when he came here to work at the Learning Center and to, in some sense, follow in my footsteps. So let me introduce you to Mike Paul, and he's going to lead you through the experiment today. Thank you, Michael.
Thank you. Hello, thank you for that wonderful introduction. I am Mike Paul. So today we are going to be doing a restriction analysis. And the restriction analysis is a really interesting lab. And basically what it boils down to is we're going to be cutting DNA, and then we're going to be looking at how DNA runs through a gel. So DNA is a very, very interesting molecule that we care a great deal about here. Cold Spring Harbor is, the home of molecular biology, and at the DNA Learning Center, we think it is really important to really make sure that everybody is understanding what DNA actually is. So in my hand, I'm holding some DNA. I'm holding a molecule of DNA. And I like to break down this model really quickly, right? So this model of DNA that I have here, it's in this double helix shape, right? So it's in the double helix shape. And the backbone of DNA, you'll notice on either side of the helix, the backbone is the same. So the backbone of DNA remains the same. And the interior portion, these base pairs, or the nitrogenous bases, they are what changes from individual to individual or organism to organism. And basically, a certain order of these bases will come together, and they will be a certain gene, and that gene will code for a protein. That protein then gives us a certain trait. So the DNA is a really interesting thing because the structure will remain the same from organism to organism, but the order of the bases is what changes, and that's what makes us different. And what is really cool, in my opinion, what I find to be really interesting and out of the ordinary, is DNA is the same structure from organism to organism. So my DNA is still in a double helix, your DNA is in a double helix, and the same 4 bases are used for me, for you, for a dog, for a tree, for bacteria, for all of these various different things. The same 4 bases are used. There's A, T, G, and C, or adenine, thymine, guanine, and cytosine. Now, these bases are so interesting. You'll also notice, if you look at my model, you notice how the red will always pair with the blue and the green will always pair with the orange? This is because adenine will always pair with thymine and guanine will always pair with cytosine. Now, the order of those bases is a gene. That gene will code for a protein. If it's in a specific order, it will code for a specific protein. The only difference between my DNA and a dog's DNA is the order of the bases will be different, therefore it'll cause different proteins to be made. So we know that my DNA is the same structure as something like this, the E. coli cell's DNA. And E. coli cells are really interesting. They are just little bacterial cells. And what they are is essentially they're just a membrane, and then inside of that membrane they have DNA, and then there's also some ribosomes in there. And basically that's all they are. It's a very simple organism. Now, the DNA inside of there is the exact same thing as my DNA. And the reason this is so interesting is because we're going to be cutting DNA and we're going to be, manipulating it to try to see if we can get everything to work the way that exactly how we want it to. So, E. coli cells and bacterial cells have developed, they have not necessarily developed, but they have evolved to have these enzymes inside of them called restriction enzymes. And what restriction enzymes do is they cut DNA at very specific locations. Now, restriction enzymes exist because of this little organism right here. This is a bacteriophage. And a bacteriophage is an interesting organism because the bacteriophage will attach itself to the membrane of bacteria, and it will inject its DNA into the bacteria. And right here I have some pictures of this actually happening. Some of these pictures are real scanning electron micrographs of this encounter actually happening. The picture on your top right is not real. That is a computer-generated image. But it is really cool because it helps to show exactly what's going on. So these little organisms attach themselves to the membrane of bacteria. They inject their DNA down into the bacteria. And then the bacteria can't really tell the difference between the bacteriophage DNA and its own DNA. Because DNA remains the same structure from individual to individual, from different organism to different organism. It's just a different order of bases, right? So it's kind of like if I have a cookbook that's a Chinese cookbook, and I have a cookbook that's an American cookbook, the structure of the book is going to remain the same, but the individual recipes inside of the book is different, and that's what makes it a different book. Now, the DNA is structurally speaking the same, but the order of the bases is going to be very different. So when the bacteriophage injects its DNA into the E. coli cell, the E. coli cell will just essentially read that DNA. It will undergo the transcription and translation, and then all of those proteins will be made. So today we're going to be working with a specific bacteriophage called the lambda bacteriophage. And it can attack E. coli cells. So the lambda bacteriophage attaches itself to the membrane of an E. coli cell. And then it'll inject its DNA in there. And then the protein-based capsule just kind of falls apart. So it injects its DNA in there, and then the bacteria will start reading that DNA and start coding for those proteins. So it starts coding for those proteins. And it starts making new bacteriophages. Makes new bacteriophages, and the bacteriophage DNA never tells the bacteria to stop making bacteriophages. So it makes more bacteriophages and makes more bacteriophages and makes more bacteriophages. And then I always like to ask this question, what happens when you put too much water in a balloon? What do you think happens? You take a balloon and you just keep pulling water in there. Just keep putting water in there. You keep putting water in there. What's going to happen? Exactly, it'll explode, it'll burst out. So it's going to pop. Now in science, what we say, we don't say explode, we don't say pop, we don't say any of that. What we say is lyse. So the cell will lyse, which is essentially burst, and all of those individual bacteriophages can now go attack a new bacteria. So all of these new bacteriophages can go attack a bacterium, and they can make this many. So this many came from one, and then each one of these can now make this many. So that is exponential growth. Bacteria, in order to survive, in order to be a successful organism in an evolutionary standpoint, they need to have a defense against this. If they don't have a defense against this, they are essentially hopeless. They need to have some form of defense. And the defense that they have evolved to have is restriction enzymes. So the bacteria, it gets the lambda bacteriophage will still inject its DNA into the bacteria. But what happens next? The restriction enzymes go and look at that DNA, they attach to the DNA, they read the sequence kind of, and wherever they see the sequence where they need to cut, they will make a cut. So the bacteriophage injects its DNA, but then those restriction enzymes just chop that DNA up. And once that DNA is cut, the bacteriophage's DNA can no longer be read as a whole, and the bacteria does not make those new bacteriophages, and it doesn't fill up, and it's not putting too much water in a balloon, it doesn't explode. It will just continue living. So this is the defense mechanism against bacteriophages for bacteria. Now, we're going to be working with two very famous restriction enzymes today. We're going to be working with EcoR1 and HindE3. EcoR1 and HindE3. Now, we have this nomenclature, kind of a convention of nomenclature, where enzymes, generally speaking, end in ACE. Now, what's very interesting is these don't end in ACE. These enzymes do not end in ACE. They end in, an R1 or a HindE3. They do not end in ACE. Now, EcoR1, the reason that it's called EcoR1 is because it was originally found in E. coli, in the RY13 strain, so that's where the R comes from, the RY13 strain, and the 1 is because it was the first one found. So the 1 is because it is the first one found in there. Now HindE3, Hind because it was in the H. influenza bacteria, D because it was the RD strain, and 3 because it was the, what do you think? The first one, the second one, the third one, the fourth one found. Which one do you think HindE3 was? Third one, exactly, thank you. It was the third one found. So the name of these enzymes will tell you a little bit about those enzymes. And it's a really interesting thing because they'll all cut DNA, but they do it at different locations. And they might do it in a different way. These two enzymes are very interesting. So EcoR1, wherever it sees GAATTC, it will make a cut in the backbone of DNA between the G and the A. So DNA is read from the 5' to the 3' end. So there's an enzyme up here reading the DNA that way. Does anyone notice anything interesting about the GAATTC? Does anyone notice anything interesting? What do you notice about that? What if this is the 5' end here, and that is the 3' end there? What do you notice? So, yes, exactly. It is repeating, or it is the same in both directions. So the way these enzymes work, one of them is going to be up here reading the DNA that way, and the other is going to be over here reading the DNA that way. And you'll notice that in both directions, it is GAATTC this way and GAATTC that way. Now what's going to happen is these enzymes are going to make a cut in the backbone of DNA between the G and the A. Now, if you look at my model, you'll notice, let's straighten this out. You'll notice that they are connected by the backbone. The individual bases are connected by the backbone, but then the base pairs are also connected. Now, the connections between the two different portions are what's really interesting to me. This is what I find really interesting. So on the backbone of DNA, the connections between this phosphate group and this sugar, those are covalent bonds. They're covalent bonds. So they're sharing electrons between two atoms. These, in the middle, the bases are held together by something called hydrogen bonds. Now, hydrogen bonds are interesting because they're not actually a chemical bond, but they're an intermolecular force. What they are is just an attraction of different charges. So if I have an atom right here that really likes electrons, it is going to kind of pull electrons towards it. And if I have an atom over here that doesn't really care about electrons, it will give its electrons away a little bit. And now with this, there's a charge distribution. So I have slightly negative, slightly positive, and those forces will attract each other. And that's what's going on here. So what this boils down to, what this really means, and what we really care about with this is the fact that on the backbone of DNA, the bonds are really, really strong. So if I break these bonds on the backbone, the bonds in the middle are just going to simply kind of fall apart. All right, so when I break the backbone between the G and the A, both sections, what ends up happening is the DNA will simply kind of fall apart. And you'll see the DNA kind of breaks up along those lines. Now, as I said before, adenine always pairs with thymine. Guanine always pairs with cytosine. So if I look right here, G is always going to be paired with a C. So A and T, A and T, T and A, T and A. So if I break those apart, theoretically I can glue them back together. I just need a different enzyme. The enzyme that I need to glue them back together is called ligase. Now, Hindi 3, the cut site is a little different. It's AAGCTT, but the cuts still happen between the A's. The enzymes still kind of read it in the same way. They'll read it just like that. And those cuts will happen, causing those to break apart just like that. Now, what I like to show you, just to show you a little bit, when I break the bonds between the G and the A, what ends up happening is these pieces just kind of move apart. Right? So these pieces just move apart. Now, yesterday, my colleague Erin, she taught a lab where we were putting green fluorescent protein into bacteria. But how do we get that gene for green fluorescent protein out of the jellyfish is the interesting thing. So it comes from a jellyfish. How do we get it out of there to put it into other organisms? And the answer to that is restriction enzymes. We use restriction enzymes to cut it out. So on either side of the GFP gene in the Achoria victoria jellyfish, there is an EcoR1 cut site. So on either side, there's a GAA-TTC. So if I take EcoR1 and I run it along the jellyfish genome, what will happen is that GFP gene will be cut out of the genome. Now I take the EcoR1 and I run it along, let's say, a mouse's genome, the genome of a mouse. I expose the fragments to ligase. What will happen is I can glue that fragment of GFP into the mouse's genome, causing glowing mice to occur. And this is a really interesting and kind of important technique. It's one of the most important techniques that we've learned. And it's impossible to do without the discovery of restriction enzymes. Restriction enzymes are extremely, extremely important. So, working with restriction enzymes and knowing how to work with restriction enzymes is what I find the most interesting portion of this. Now, what we're going to do is we're going to cut DNA today, and we're going to cut DNA in a very specific way. We're going to do it in a way that we know how it's going to be done so that we're ensuring that everything will be done correctly. So if I'm cutting DNA, what's one thing you think I'm going to need? What do you think? Don't overthink it. Yeah, DNA, awesome. So if you're working with DNA, you're going to need to cut DNA, and you're also going to need enzymes. So you're both right. So you're going to need DNA, and you're going to need enzymes. Now, my question for you is, do enzymes just work at every pH? Are they going to work really acidic, really, really basic? What do you think? No, they don't, right? They need to be at a very specific pH. Does anyone know how we make a solution stay at a very specific pH? What do we use? Yes, buffer. Awesome. So we use a buffer. Now the buffer that we're working with today is a very specific restriction buffer, and that buffer is set to 7.4 is the pH that we're working with. Now, the pH needs to be controlled. What else needs to be controlled for enzymes to work? Temperature. Awesome job. So the temperature also needs to be controlled. So pH and temperature. The temperature that we're working with today is 37 degrees Celsius. What is the significance of 37 degrees Celsius? What do you think? People. Yes, it's human body temperature. So that is a really interesting thing. These enzymes work best at human body temperature. And that is not really surprising when you think about, what do we have? We have a lot of bacteria inside of us, right? It's always a fun thing that I always like to say, we're mostly bacteria. We have more bacterial cells in our body than human cells. which is just an interesting little fun fact I always like to bring up. Now, so basically what we said is we need DNA, we need buffer, and we need enzyme. And that's all we really need. So the first thing that we're going to do, whenever you're doing an experiment like this, we need to label our tubes. So I have 4 tubes up here, 4 tubes. And they are labeled in a very specific way. I have a tube labeled E, H, a minus, and a question mark. Now, in the E tube, I'm going to cut lambda DNA with eco R1. In the H tube, I'm going to cut lambda DNA with Hindi 3. In the minus, I'm not going to cut the lambda DNA at all. What is that? Why do I have that? Why do I need a minus tube where I'm not cutting the DNA at all? What's that called? Control group, yes. So in order to know that I cut the DNA properly, I have to make sure that the DNA looks the way I expect it to when it's running uncut through the gel. It's a really important thing. So then I'm also, just for fun, we're doing a mystery tube, and the mystery tube could have eco, it could have Hindi. You're going to tell me at the end. It's going to be eco R1 or Hindi 3, and you're going to tell me what it is at the end. It's going to be really fun. All right, so basically, what we need is we need each one of our tubes to have 5 microliters of buffer, 4 microliters of DNA, and then 1 microliter of whichever enzyme or water is going into the tube. But you'll notice when we're looking at this matrix, every tube is going to need buffer, and every tube is going to need DNA. So every tube is going to need buffer, and every tube is going to need DNA, so that is what we're going to add first. Now, if you look at this, it has this little symbol in front of the liter symbol. It has that symbol in front of the liter symbol. Does anyone know what that symbol means, what that prefix is? What does that stand for? It's a Greek letter called mu, and yes, micro. So it is the microliter. Now, a microliter is 10 to the negative 6th of a liter. Now, I always like to put this into some form of context, right? So you take 1 liter, you cut it into 1,000. What do you get? 1 liter, you cut it into 1,000. What do you have 1 of if you take 1 of those pieces? A milliliter, exactly. Now you take that milliliter, you cut it into 1,000. Then you have 1 microliter. So 1 microliter is 1/1,000,000th of a liter. It's 1/1,000,000th of a liter. It's a very, very small volume. Do you think I can measure this volume just in a graduated cylinder with my eyes? What do you think? No, definitely not. So I can't measure this with just my eyes. I need a special tool. And the special tool that I use is this, a pipette. Now, this is a micropipette, and it's a very awesome tool. This one is set to measure from 0.5 microliters to 10 microliters, from 0.5 to 10. So, what I need to do is I need to set it to measure 5 microliters. The way that I do that is I twist this knob. You'll notice the numbers on my screen are changing. Oops, went a little too far. It's set to 05 line 00. Now this is set to 5. You'll notice that line right there, that is the decimal point. So this is set to 5 microliters. Now, I'm almost ready to go. What I need, one other thing that I need for this experiment is just a tip on the pipette. All right? So the very first thing, if I have it set to 5 microliters, what I'm going to add first is the buffer. So in this tube right here, I have my buffer. Oops, sorry, nope. In this tube right here, I have my buffer. All right? So I am going to push down to the first stop. You'll notice if I push down lightly, I go to the first stop. If I push down hard all the way, I go all the way down. The first stop pushes out 5 microliters of air. So if I take my tip, I go into the liquid and I release, I'm going to pick up 5 microliters of liquid. So down to the first stop, into the liquid, slowly release. I like to check to make sure that I have liquid there. Then I open this tube, I go down to the bottom of the tube, press against the side, down to the second stop. Hold my thumb down until I am out of the tube. All right? Now I need to do that for all 4 tubes. I'm going to add 5 microliters of buffer to all 4 tubes. So the E tube, the H tube, the minus tube, and the question mark tube. Now, what I'm doing here is I'm helping to make sure that my pH will be controlled during this experiment. So that's the point of the buffer, and it's good to just kind of remind you that. Now, I added the buffer to all 4 tubes, so the buffer tube can then be thrown out. I have a little waste speaker right up here. So I added the buffer to all 4 tubes. Then I'm going to change my tip, just press down on this, and that tip will shoot right off. What do you think the next thing I need to add is? If I added the largest volume to all 4 tubes, what do you think the next thing I need to add is? What would make the most sense? The DNA. Awesome job. So the DNA needs to be added. But I need to change this volume, correct? So what I do is set it to 4 microliters by changing it to 0,400. 0,400, just like that. Now, it's the same thing. Once again, I grab a tip. I grab a tip, down to the first stop, into the liquid, slowly release. I like to check to make sure that I have liquid in there. Then I'm going to inject this liquid into the liquid that is already present. Now, I have buffer in each of these tubes, and I have DNA right here. And since I'm just mixing the DNA and the buffer in all of the tubes, I don't need to change tips quite yet. So down to the first stop, back into the DNA. Now that would be different if I was doing an experiment where I am going to reuse this tube for multiple different things, but since I am just mixing DNA and buffer and then I'm going to throw that tube out, we can just use the same tip over and over again. Now, I have a question for you. Why do I need to change my tip ever? Why is that an important thing? What will happen? To wash. Well, yes, exactly. Contamination, awesome job. So if I'm working with EcoR1 and I don't want to work with Hindi3, I can have an EcoR1 tip right here, and then I'll have a Hindi3 tip at a different point in time. All right? So now I've added the DNA, I added the buffer. It's time for the enzymes. Now the enzymes are really difficult. This is where we start to get into the point where we need to really be careful about our tips. So the enzymes, it's 1 microliter. 1 microliter only. So I'm going to change the volume down to 1. So 0,100. Now what I want you to notice about the pipette is the button is significantly smaller. If I change this, the button will go really, really high. If I go back to 1, the button will be really, really small. Now, this is an interesting thing because it makes it a little bit more difficult. To pipette. So you'll notice that the difference between my first being completely released and pushing down to that first stop is really, really small. The difference between being completely released and the first stop is really small. So we have to be very careful. All right, so set to 1 microliter, I grab a tip. I grab my Hindi tube, my Hindi3 tube, push down to the first stop, go into the liquid, I release, I check to make sure there's liquid in there. Now, I have to be very, very careful that I'm adding it to the proper tube. So this tube I labeled H. I need to be very careful that I'm adding the Hindi3 to the H tube. All right, so I go in there, down to the second stop, hold my thumb down until I'm out of that tube. I can get rid of this tip. Now, I changed my tip, so I'm all set to move on to the next enzyme. The next enzyme is going to be EcoR1. I'm going to add that one into the E tube. To grab that, I go into the E tube. Then, you guessed it, have to change your tips. Next one, the water. Going to take this down into the water. I grab a microliter. I put that into the minus tube right there. And finally, the question mark tube. This is the mystery, and it's kind of the most fun portion of this experiment. At the very end, you'll see, we're going to see a couple of gels today. At the very end, you're going to tell me whether or not what my question mark was. Whether it was an Eco, whether it was an Hindi3, whether it was nothing, or whether it was a different enzyme that we don't know about. Now, earlier I said we're going to need these enzymes to be at 37 degrees Celsius in order for them to work, right? So I'm going to take these tubes and I bring them over to my water bath. This is set to 37 degrees Celsius. I'm going to take my 4 tubes and put them into 37 degrees. And this is just the optimal temperature for these enzymes to work. I'm going to let them sit in there for a little while and let that cutting occur. While I'm doing that, I'm going to ask you a question. Can we simply see DNA? If you're just looking at it, can you just see DNA? No, right? No. Now, there's a couple of interesting things. There's a couple of interesting things that we need to do. First thing, we're cutting DNA, correct? So we're going to be cutting DNA. Now, well, yes, PCR can let us see, definitely. But what we're going to do is we're going to be cutting DNA. And we want to make sure that we can see that we're cutting it in the right area. So we're going to have to, one, use a tool to see the DNA, two, use a tool to separate out the DNA. Now, DNA is clear and colorless, right? So it's colorless. What I need to do is give it some color. And for that, I'm going to use a loading dye with this little dye on it called cybergreen. Cybergreen. Now, I'm going to add this to my tubes. And then, I need to separate out the DNA. And the way that I do that is through a process called gel electrophoresis. We're going to talk a lot about gel electrophoresis, but before we get into that, the very first thing needed for gel electrophoresis is an agarose gel. An agarose gel. Now, earlier in the lab, I said enzymes end in ase. Agarose ends in ose. Does anyone know what type of molecule agarose might be? If it ends in OSE as opposed to ASE. What type of molecule is agarose? What do you think? So this is a trickier question. Sugar, yes, it is a sugar. So agarose is a sugar that comes from a seaweed. The seaweed's called agar-agar. It's highly purified, and it is just this sugar. And what we do is we put this sugar into an ionic solution. And the solution is called TBE. And this is a mixture of tris base, boric acid, EDTA, and NaOH. And basically, it's just an ionic solution. It's going to help conduct electricity, which you'll learn why that's so important as well. So to make our gels, it's kind of like making Jell-O. When you make Jell-O, the Jell-O is really, you have to get the water really, really hot in order to, make it a liquid, and then when it cools down, it will still solidify into that gelatinous form. Agarose is the same thing. So, I'm going to make an agarose gel. I'm going to put this stopper on there. I'll put this stopper right there. And then, I'm going to put that down. Now, I'll take my comb, because eventually I'm going to need to put the DNA in there, and for that I have to have this comb. So I'm going to put that comb in on these black notches right here. So I put that comb right there, and you'll notice the comb has a little black line on it. That black line is going to tell me where I need to pour to, okay? So I'm walking over here and I'm grabbing the agarose that I have. Now the agarose is fairly hot because it needs to be in order to keep it a liquid when I'm pouring it. So I take this and I'm going to pour it in right up until the black line. So I'm going to pour it until it goes to a black line. Ready? And that's perfect. So now I'm going to let that sit and I'm going to let that solidify. And what's happening is the agarose molecules are large polymers. So the agarose molecules themselves are large polymers. And individual polymers are starting to interact with other polymers. But when it's hot and when it's a liquid, those molecules are moving too fast for those interactions to occur. Once it starts to cool down, those molecules slow down, those interactions between the individual polymers start to form, and the gel starts to solidify. All right, so the gel will start to solidify. Once the gel is solid, we'll be able to run DNA through there. Now, in the interest of time, I have a different gel right here that I poured, and it is already solidified. All right, so this one is already solidified. So I'm ready to work with this one. Now, I need to get these bumpers off. And the bumpers are pretty interesting. They kind of peel away. So they'll peel away on this side. They'll peel away on this one. Then I need to take this comb and pull it straight up. Get that right out of the agarose. Now, eventually I'm going to be conducting electricity through this gel. I'm going to conduct electricity through the gel. Now, electricity doesn't just fly through the air. We need to get this in a solution that will help to conduct the electricity through the gel. So in order to do that, I'm going to use this little gel box right here. Now, I'm going to open this gel box, and inside of it, you'll see there's a black line and a red line. this will tell me which side of the gel is getting which type of charge. So I need to make sure that I line the black line on my gel tray and the red line up with the black line and the red line in the gel box. Now, as I said earlier, the electricity is not going to just fly through the air. It needs an ionic solution in which to kind of conduct the electrons across the gel. And this is the TBE. So this is an ionic solution with, the tris base, boric acid, EDTA, and NaOH. So I'll pour that in right here. I pour it in on one side until it covers the gel. And then I do the same thing on the other side. All right, now, so currently I am ready to load my gel. What I'm going to do, before I load my gel, we're going to talk a little bit more about how Agarose and how gel electrophoresis actually works. Now, what electricity is essentially just a flow of electrons across the potential. Now, this technique, gel electrophoresis, was discovered at Cold Spring Harbor Laboratory. And it was discovered by Phil Sharp, Joe Sambrook, and Bill Sugden. Now, what they use this technique to do is to really separate out the DNA molecules. They can use this technique to separate out individual fragments of DNA that are 100 nucleotides in length to over 10,000 nucleotides in length. And what happens is the gel is submerged in a liquid that will conduct electricity. Now, electricity is simply just the flow of electrons across a potential, right? So it's just electrons flowing. Now, electrons are little negatively charged particles. So one of the wires is going to have a buildup of negatively charged particles, and the other is going to have a dearth or an absence of these negatively charged particles. And that's what's going to make it be positive. So what do you think? Is DNA negatively charged or positively charged? What do you think? Negative. It is negatively charged, yes, so it's negatively charged. And what we're going to do is we're going to line the DNA up very close to that negative charge. So if I have a negatively charged wire right here, I line the DNA up right there, what's going to happen? Is the DNA going to be attracted to the negative? Is it going to be pushed away from the negative? What do you think? Attracted. Not necessarily attracted. So the like charges will get it. Yes, exactly. They're going to push them away. The like charges will repel, and it's going to cause the DNA to be pushed into the gel. It's going to cause the DNA to be pushed into the gel. Now, what's going to happen is we start that electricity up. We put in our samples. So we take our samples, we put them in, we get that charge going, and once that charge starts to move, the DNA will start to move just because of that negative charge. So the DNA starts to move through the gel. And this will show you how the DNA moves through the gel. So the larger pieces are going to have a very difficult time navigating their way through that gel matrix, right? So the agarose is forming this polymer matrix that's going on. And the large pieces of DNA, as they try to get through the matrix, are going to have a really difficult time. They're going to bump in all the other molecules of agarose as they're trying to move forward. But a small piece can just fly right through. Now, this is interesting because it's the exact same way that you would expect it to be, right? If you see a small motorcycle and you see a large truck trying to get through the same traffic jam, the motorcycle is going to have a way easier time than the large truck. So this is the way that this works, right? So the small pieces are able to move right through while the large pieces are getting hung up every time they're trying to move. All right, now, our gel is pretty interesting. Agarose is really fragile. So if I take my pipette tip and I stab it down in there, what's going to happen is I'll rip the agarose right apart. I don't want to do that. It's a bad thing. So what we're going to do is we're going to load in a very specific way. We're going to load it in a very, very specific way. Now, I have 4 samples and I have 8 wells. So what we're going to do is we are going to leave these 2 wells and these 2 wells open, and we're going to load in those 4 central wells. And we're going to load it as the E tube, the H tube, the minus tube, and the question mark tube. Earlier, I said that we need to add this loading dye to our samples because DNA is completely see-through. It's like clear and colorless. So I need to add that in order to see everything that's going on. So what I'm going to do is I pick up my tubes from over here. I grab my 4 tubes, put them right down, and I need to add 1 microliter of Cybergreen to each one of those tubes, Cybergreen loading dye. Now, the loading dye is great for multiple reasons. First one is the Cybergreen. Cybergreen is a dye that will go into the DNA, go in between the base pairs. It's a dye that goes there. And this dye will fluoresce green. So if it attaches to the DNA, it will make this fluorescent pattern that makes it look green when I expose it to ultraviolet light. So it helps the DNA glow. Now, the loading dye also has some sugars in it. One, to make the loading dye dense, it'll fall down to the bottom in my buffer, it'll fall down to the bottom. The other reason is it has 2 different sugars in it. And those different sugars are different sizes. So one's going to have a way easier time of moving through the gel than the other. And this will help track where my DNA is. So the 2 sugars will run through the gel. Now, I add 1 microliter of Cybergreen to each of my tubes. I open this up. I go in here, pick up a microliter. I take that microliter and I inject it into the liquid that's already in the tube. Now, the one thing that I want to tell you guys about Cybergreen is it is a dye. So this loading dye is a dye. And I have to say this because if you ever do this experiment at home, or if you ever come here and you're doing this experiment, I want to make sure that you guys are careful when you're working with this because it is a dye, so if it gets on your clothes, it will stain. If it gets on your hands or face, you'll be walking around with blue hands or a blue face for a little while. You notice that I'm changing the tips every single time. Just one more time. Why am I doing that? Why do you think I'm doing that? Why do I need to change the tips every single time? Yes, contamination. I don't want to get any eco in my Hindi. I don't want any of the other tubes to get in my question mark. I don't want anything to mix with anything at this point. So I need to change my tips every single time. All right, so I'm adding the Cybergreen to all of these tubes, and now I am ready to load my gel. So we're going to load a full 10 microliters. And the 10 microliters is going to be set on the pipette just like 1, 0, 0, 0. And you'll notice that the button gets really tall at this point. So I grab a tip. Grab my E-tube. That's the first one that I'm going to work with. I need to get this into the wells that I created with my comb. So the way I do this, I push down to the first stop, I go into the liquid, I slowly release, I pick that liquid up into my pipette. This tube can now be thrown away. What I do is I'm going to take my dominant elbow, put it down on the table. I'm going to take my non-dominant hand and touch right here. Now I need to make sure that I am loading in the right well, so I get right over it, and then I am going to break the surface of the liquid, just break the surface of the liquid right above the well, and then I'm going to push down to that first stop to load the gel. Hold my thumb down until I'm out. Now, I did a couple of things there. The first thing, I just broke the surface of the liquid, right? I didn't actually go down into the well. That's what we would think. That's what it looks like to me. The reason it looks like that to me is I'm actually in the well when I just break the surface of the liquid. Due to the refractive index of water, when I take the tip and I put it into the liquid, it looks like it's very shallow. It looks like it's just right there, but it's actually deeper and it's actually going into the well. So if I look like I'm trying to go into the well, I'm going to actually stab the bottom of the gel. So you want to just break the surface of the liquid, push down to that first stop only. Now, can I just instantly go into my next tube? What do I have to do? What do I have to do? No, I can't. I need to do something. What do I need to do? Change the tip. Exactly. So I need to grab a new tip, and then I grab a new one, and now I did the E, so the next tube that I'm going to do is the H. Down to the first stop, into the liquid, I slowly release. I can throw that tube out now. Now, you'll notice that when I'm doing this, I'm only pushing down to the first stop. I'm no longer pushing all the way down. The reason that I'm doing that is when I go down to the second stop, I'm going to release an air bubble. And I don't want to put an air bubble under my sample, so I'm only going down to the first stop when I'm loading the gel. Down into the well, I push down to the first stop, and I hold my thumb down until I come out of the gel. if you're ever doing this here, if you're ever doing this at home, one thing that will tell you that you're doing a great job is if you get perfect little rectangles that seem to match the gel perfectly, that seem to match the comb perfectly, that will tell you that you're doing everything the right way. All right, so next one is minus. Once again, I am just breaking the surface of liquid right above the well, and then I push down to the first stop, hold my thumb down until I'm out. Change my tip, and finally, this is my question mark time. Down to the first stop. All right, now. I need to conduct electricity across this. So, what I do is I'm going to connect these electrodes right here. I connect the electrodes onto the gel box. Now, I need to connect these to a power supply. So I take the red and I match it to the red area right here. I take the black electrode and I put it in right there. Which of these two, if my DNA is closer to the black wire, which of those two is negatively charged? Yes, the black tube is negatively charged. Now, I actually just noticed a question asking if the DNA will mix when I inject it in there, and the reason it won't actually do that, and the reason is because the individual lanes will stay separate from each other. So the lanes will stay separated from each other. Now, I set my power supply up to 140 volts, and I did that for 30 minutes. So what I'm going to do is click start. And once that's starting, the electricity is building up near the black wire, and it's pushing that DNA. It's going to push that DNA in a line. So the DNA that's in one well is going to stay in line with that well. Now, if I have a really, really small piece of DNA, it's going to move very, very quickly away from that well. If I have a really large piece of DNA, it's going to move very slowly away from that. And that's how the separation occurs over time. So I'm going to let this gel run, and what we're going to do is when this gel is running, we're going to look back up at my screen. So, lambda DNA is 48,502 bases long. 48,502 bases long. And in that 48,502, there are 5 distinct locations of GAA-TTC. 5 different locations. 1, 2, 3, 4, 5. There are 5 different locations. Now, if I cut a piece of string 5 times, how many pieces of string do I get? How many pieces? I cut it 5 times. I get 6 pieces, exactly. So if I cut it 5 times, I should get 6 individual strands of DNA, which means I should get 6 different bands in my gel. But the thing with ECO is I don't. I actually get 5 bands. And the reason, it's really 5 bands. Now, the reason it's 5 is because this fragment right here is 5804, and this fragment right here is 5643. Now, 5804 and 5643 are simply too close in base pairs to each other to really separate out during the time that I'm running. If I wanted to get those two to separate away from each other, what could I do? What is one thing that I could do to get those to separate out? What do you think? This is a tricky question, right? So if I need those two to separate out, I'm pushing them with the same force. I don't do a PCR. Don't do a polymerase chain reaction. I run the gel longer. Awesome job. Yes, I run the gel longer, and that will help to increase the time when it's running. And over time, the difference will start to become larger, right? So it'll start from being really close to each other, and I've run it longer, and I run it longer, and I run it longer, and I run it longer. The separation starts to occur a little bit more. All right, so Hindi 3, let's focus in on that one. Now, Hindi 3 gets cut 1, 2, 3, 4, 5, 6, 7 times. 7 times, 7 cuts should result in how many fragments? 7 cuts should result in how many fragments? 8. Should result in 8. Now, the interesting thing about Hindi 3 is for the most part, there's one fragment right here that's 125 base pairs long. And that's always going to be the case. When I cut lambda DNA with Hindi 3, there's always going to be one fragment that's 125 base pairs long. Now, 125 base pairs long, simply too small. With the length that we run our gels in order to get separation for the top bands, that 125 just runs right out of our gel and goes right onto that red wire. So it runs right through the gel. The 564 fragment right next to it, that one, is also an interesting thing. We do see it sometimes. However, it's simply so small. There's only 524 areas where the cybergreen can really bind in there. So it doesn't create a strong fluorescent signal. So if it doesn't create a strong fluorescent signal, what happens is we don't have enough photons for it to register to our eyes that there's actually a piece of DNA there. So we need to, in order to see it, which you can see it sometimes, what you need to do is you need to make sure that you're working with kind of a higher concentration of DNA. You need to make sure that your gel is kind of thin, and there's a very specific set of things that need to happen in order to see that band. So let's actually look at a couple of gels here. I have two gels. I have my EcoR1 right here, and I have my Hindi3 right next to it. Now let's actually zoom in on Hindi3 for a second. Let's actually zoom in on that, and let's look just as this one lane right here. Now, Hindi3 is really, really interesting because if I go back, I said there could be 1, 2, 3, 4, 5, 6, 7 fragments if I see that 564. That 125 one's just running right off the gel. So there is going to be 1, 2, 3, 4, 5, 6, 7 fragments. The largest fragment should be the closest to the top of the gel. So when I look back over here, I'm going to see that the largest fragment of the Hindi, right here, 23,130 base pairs long, is right up at the top of the gel where it should be. The next line is the next biggest fragment, and the next biggest fragment, and the next biggest fragment. And then these two are smaller, and then the smallest one, 564, is very, very faint, and it's all the way down there. This shows me that the larger pieces are moving slowly through the gel, while the smaller pieces are moving really quickly through the gel. The well that we loaded would be right up here. All right? So if I go back and I look at my Eco lane, I look at my Hindi lane, the Eco has 5 bands, right? 1, 2, 3, 4, 5. This central band is the band that is the 5,643 and the 5804 running together. That's the central band. But Eco, if I have 5 lanes, that means it's cut perfectly. So that one did a great job. Now the Hindi, 1, 2, 3, 4, 5, 6, and you can see the very faint 564. That one, perfect job. The minus, it is 1 band. Now you'll notice the minus being 1 band, but it has these long streaks coming out of it. It has those long streaks, these ones right up here. Those streaks are caused because the lambda DNA is 48,502 bases long. It's simply too long. So it'll basically get caught up and cause it to be streaky. It's like over capacity, it's over the capacity of the actual agarose itself. So it starts to kind of streak out a little bit. Now, what is my question mark on this gel? Is my question mark EcoR1 or is my question mark Hindi3? Yes, EcoR1. EcoR1 is what the question mark is. Now, if I look at this gel over here, is my E lane good? What do you think? What do you think? Is my E lane good? Yes or no? So the E lane is done. Yes, it's done perfectly. My Hindi lane, is it done perfectly? Yes, it is done perfectly. My minus lane, it's done perfectly. And what is in my question mark? EcoR1 or Hindi3? Hindi3, yes, Hindi3. Awesome job. So this is what we call the ideal gel. What I want to do is I want to show you what a student gel looks like sometimes. So let's go all the way to the end. And this is an example from a class that I have recently taught. This is an example from a class that I taught right down the road at Harbor Fields High School, right down the road from Cold Spring Harbor. Now, this gel is interesting. You can see the Eco is a little bit brighter, the Hindi is a little bit darker. Some mistakes can happen when you're teaching a young group of kids, but basically, you'll see this has 5 bands right there. This one has all of the Hindi bands. The minus is perfectly done, and the question mark was what? Eco or Hindi? What do you think? Yes, Eco. Perfect. All right. So I just wanted to say that's pretty much everything for the restriction analysis. Now what I just wanted to say is thank you so much for taking the time to kind of watch this video. If you liked what we did here today, we really hope to see you at some point in time. So if you ever want to come to Cold Spring Harbor, please, come on over. We would love to have you. Also, just wanted to say, I hope everything is going as well as it possibly can with the coronavirus. I hope that everyone is having as good of a time as they possibly can. Now, before you guys go, you just saw a poll pop up at the top of your screen. What I would like you to do is just answer those questions. So question 1, in which school are you currently enrolled? And just click submit when you're done. We're gonna have a second, just to answer it, and then we'll close it out. So you guys just answer that poll. And I can also answer questions. I have a couple of minutes, so if you guys have any questions, you can type them out. All right, so we're just taking a little time to answer that first one. How long are we going to run the gel for? So we run the gel for 30 minutes currently, and that's just going to help separation. If I want to run it for longer, I can. It'll separate things out a little bit more. So I can, run it for longer if I want to get more separation, or I can run it shorter if I don't care that much about that. All right, so if anyone just answer that first question and just click submit. All right, so here are the results from our first question. We have 25% of you from Beijing, number 166, and a really nice widespread of a lot of different people, a lot of different schools there. All right, so let's go to the next one, and the next question is, which grade are you in now? So you guys just answer that and submit it as quickly as possible. Just have it's a total of 3 questions. Why is DNA negatively charged? DNA is negatively charged because of the phosphate group. So DNA has a phosphate group in it, and actually, we can go here, and we can actually take a look at what the phosphate group is. So after you guys answer that, we can look at this screen, and we can look at the phosphate group, which is a portion of the DNA. That's why it's an acid. Yes, that's why it's an acid. Yes, so the phosphate group right here, it's a PO4 group. Oops. It is a PO4 group, and basically those oxygens are going to have a negative charge. And now it has a permanent negative charge because oxygen's really good at stabilizing a negative charge. So we just saw that most of you guys are from 5th through 8th grade, which is awesome. Love to see that. And the last question is coming up. So if you guys take a second and just look at that, this right here is that PO4. Would you want to come to the DNA Learning Center after watching that video? That's the question. Hoping to get 100% yes on that, please. I don't have a YouTube channel, I saw that. But the DNA Learning Center does, correct? Yes. 94% yes, all right, that's good. All right, awesome job. Thank you guys so much once again. I hope everything is going really, really well for you. From Cold Spring Harbor, we hope to see you one day. So thank you so much.